So once you understand how normal cell division of this type, cell cycle, happens, then we can start to think about ways in which things can go wrong. First, I'm going to show you what we call the normal checkpoints of the cell cycle, and then we'll talk about what happens when they don't work. So there are three important checkpoints in the cell cycle that are known. One is found between or right at the end of G1 and before S phase begins, and that's called the G1S checkpoint. Probably the most important checkpoint, really. This is where if a cell goes past this checkpoint, it means the cell is committing to the cell division. Because in G1, a cell can decide not to divide and go into a phase called G0. So if a cell gets past this point, it's making a commitment to divide. And this is where there's often signals from outside the cell called growth factors that are required to make the cell choose to divide. But a lot of cells will go into a G0, which is just they're just living, they're not dividing in that phase, phase. The next checkpoint is called G2M. It's at the end of G2, right before M, and the cell is making a commitment again to going through mitosis, karyokinesis. And at this point, the cell is surveying, we call it surveillance, whether or not the DNA replication is complete and anything that was damaged, any DNA problems have been fixed. So it's an important surveillance of the genetic material that goes on there. And then in the middle of the M phase, specifically in metaphase, there is a checkpoint in which the cell is checking to make sure each chromatid is connected to a spindle fiber so that the chromosomes will be segregated properly. So these are the three main checkpoints. And at these checkpoints, the cell cycle, if the cell isn't able to kind of meet the requirements, the cell can stop and delay moving forward, and it can fix the problem. However, if the cell cannot fix a problem, let's say a cell gets stopped at a checkpoint and it tries to fix the problem, but it can't, what will happen is the cell will initiate what's called apoptosis, apoptosis. All right. And apoptosis, sometimes pronounced apoptosis, sometimes the P is considered silent. It just depends on who you talk to. But this is what we call a programmed cell death. So the cell will commit suicide if the cell is not able to correct the problem that might be there. And so the cell has to either correct the problem and move forward through the cell cycle or go into apoptosis. That's the normal sequence of events. And so here we have that same pie diagram, that same clock diagram, but now they've added the checkpoints. So right about here in terms of time is where the cell is checking to make sure everything is ready to go into S phase, or the cell can sort of off-ramp here into that resting state called G0. So if the cell decides to move forward with cell division, it'll go into S phase. And then a cell that has gone through S phase then has another check here at the end of G2 to make sure the DNA everything's been replicated, everything's been fixed if there's been any damage, and it goes into M. And then in the middle of M phase, the cell is checking to make sure the spindle is attached to each chromatid. So that's the visual of those three checkpoints. So I mentioned that G0 is the phase that the cell can go into, and it's considered to be sort of an off-ramp from G1. So if the cell is not going to be dividing, if it doesn't get the signals, the growth factors that are required, the cell will stop and go into G0. So it says dormant. It just means not a dividing cell anymore. The cell is still living, it's still doing cell respiration, and it's still moving, moving proteins and making things, but it's not actively in a cell cycle. And some cells in your body will stay in G0 the rest of your life, and they'll just die naturally. And other cells can be in G0, but then be stimulated later to go back into a cell division. So it depends on the cell type. Typically, neurons and muscle cells will never, once they go into a G0, they won't divide again, but other cells can. So it depends on the type of cell, honestly. But G0, a cell is living, it's fine, it's just not dividing. So let's think about cancer. Cancer is fundamentally a failure of cell cycle control. And if the cell is not able to control its cell cycle, then it results in an unrestrained, uncontrolled cell division. So what you start with one cell and it starts to lose control. And the loss of control is typically from gene mutations, because there are genes that make the proteins that control the checkpoints. So if the genes get mutated, then the proteins controlling the checkpoints are not normal. They're not functional. And so when a cell no longer has proteins regulating the checkpoints, then the cell just goes through the checkpoints and doesn't fix any problems and doesn't scan for DNA damage, and it doesn't check and make sure that the spindle fibers are attached properly. And so all of these things start to go haywire. And the cell will continually divide and divide and divide. And so when you end up with a clump of cells that are all from the division of one original cell that keeps dividing uncontrolled, we call that a tumor. And really there's two kinds of genes that we have that, if they get mutated, will typically start the process of forming a tumor. And those two categories of genes are called proto-oncogenes and tumor suppressor genes. So we're going to go through a little bit about proto-oncogenes and a little bit about tumor suppressor genes. Okay, when everybody has proto-oncogenes. These are normal genes in your cells, and we call them proto-oncogenes because they're normally controlling the checkpoints. They're encoding the proteins which encode the checkpoints, or which control the checkpoints. But when those genes get mutated, we change the name and we call them oncogenes. And oncogenes means cancer genes. And so you only need one mutated proto-oncogene that becomes an oncogene to start that one cell into an uncontrolled cell division. And all tumors start from just one of your cells in your body that gets these kinds of mutations or loses control. There is a gene we all have, it's called RAS, and about a third of all types of cancer or tumors will have a mutation in that particular gene. So that gene is very commonly mutated in all different types of cancer. And by types, I mean like pancreatic cancer and breast cancer and ovarian cancer and prostate cancer. So a lot of types of cancer, colon cancer, have mutations in RAS. The other type of gene that can be mutated to start a cell into this process is called the tumor suppressor genes. The most famous tumor suppressor gene, arguably, is called P53. P53 is important at the G1S checkpoint, and if it gets mutated, then it doesn't function properly at the G1S checkpoint. So normally a cell, if the DNA is damaged, it can stop and repair the DNA. But if the DNA is not repairable, then P53 will direct the cell into apoptosis. But if P53 is mutated, then the cell can't go into apoptosis, and then the only option is just to keep dividing uncontrolled. So it's just a doozy of a mutation. about half of all types of cancer will have a mutation in the p53 gene. So what we're saying is that a group of cells called a tumor will have the mutations. The rest of your cells don't have those mutations. But it doesn't matter because the tumor can spread and create all kinds of problems in your body. And so the first tumor suppressor gene that was ever discovered, called RB, was discovered at MD Anderson in Houston, and that's still a very big research center for all of cancer research in the world, is at MD Anderson in Houston. In this picture, I just wanted to show you there's actually a lot of genes that are important for signaling these cell checkpoints, and any one of them, if it gets mutated, can cause that cell to start on that path of forming a tumor, being cancerous. You do not need to memorize this or anything, but there's this very complicated series of events that involve many genes and proteins. And so it's actually surprising to me sometimes that we don't have cancer more often, because there's so many genes that are involved in keeping the cells normal in the terms of the cell cycle. So so that is all I have for you guys.